Hey, Dr. Manella, welcome to my final e-report. The cell membrane, also called the plasma membrane, is a fatty, thin film that confines life. It accepts or declines entrances and exits to serve as either a portal or a barrier to the outside environment. Lipids, specifically phospholipids, create that barrier. Proteins create the portals that serve as either pumps or channels, and the membrane can shape, stretch, flex, and grow without tearing. But when it does tear, it is also self-healing. The plasma membrane confers functions through proteins and fats, which give it its specific abilities. The membrane is made in the rough ER out of many phospholipids. Phospholipids have a hydrophobic tail made of hydrocarbons and hydrophilic heads containing charged groups. This makes them amphipathic. That drives spontaneous membrane formation. Hydrophobic tails force lipids to clump together at the heads to prevent water from reaching the tails. This creates this micelle structure. One lipid tail contains a nick caused by a double bond in the hydrocarbon tail. This nick prevents the lipids from forming that initial micelle structure. Therefore, lipids form in a secondary structure where they pack tail to tail like this. This is a liposome. It allows for effective clumping at the head so that the water does not enter the tails. This spontaneous formation is how we get the phospholipid bilayer. Those two opposing phospholipids connected by their heads and touching at their tails make up the cell membrane. Lipids can't have edges exposed to the water, which makes them want to round out. This allows for self-healing. Phospholipid bilayers create a finite sheet free of edges. Here the lipid bilayers roll itself into a ball to conserve energy. The bilayer creates a two-dimensional fluidity where hydrophobic regions cannot exist happy in a fully aqueous environment. Lipids can't pop out to move once they are sealed, so they use lateral and transverse diffusion to move. Lipids and proteins can float moving side to side but not up and down. This is 2D fluidity, and that's in lateral diffusion. The components of the bilayer can flip over once a month called transverse diffusion. But the membrane is asymmetrical, and the inner and outer face of the membranes contain different phospholipids with different protein components. The phospholipid heads of the inner layer are smaller, while the ones on the outer layer take up more room. This creates a natural curve in the bilayer, which ultimately creates this full sphere. When transverse diffusion occurs, it must be fixed. So, that's when flipase comes in and will reverse the flip. Membrane proteins are responsible for the activity and function of the membrane. They make up about 50% of the cell membrane with a main function of transportation. Integral proteins are anchoring embedded proteins and can only be liberated by a detergent. They link intracellular actin filaments to extracellular matrix proteins. A type of integral protein is a transmembrane protein, and that is threaded between two domains. The transmembrane proteins serve as a protein bridge between an intracellular and extracellular component. Peripheral proteins are found on the periphery where they do not cross the membrane, but they can be attached to either side of the membrane. They have several functions such as transport, signaling, and maintaining the cell shape and structure. Receptor proteins hear extracellular signals and generate them into an intracellular signal that causes an intracellular response. The cell cortex is a durable structure part of the plasma membrane, since it's actually flimsy. It is a meshwork of proteins lying underneath that connect to the cell membrane. Membrane domains allow compartmentalizing. They are necessary because membrane proteins occupy specific regions of a membrane. These regions are specialized. The formation of these domains are either 1. The membrane sticks to something that lies outside, 2. The membrane protein sticks to something inside, or 3. Structures in or associated with the membrane that restrict protein movement floating past a specific region. A transporter is a protein that contains moving parts and can transfer molecules from one side of the membrane to the other. This happens through change of shape, where they can move molecules in or out, but not in either direction against their concentration gradient. Channels are proteins with pores with no moving parts. They make hydrophilic holes in the membrane that select molecules can use. Usually these molecules are ions, so they are called ion channels, where ions also move with their concentration gradient. The positive charge must be regulated by anions. Ions, because of their charge, do not diffuse across the membrane, and they are moved through the membrane. Charged molecules are hydrophilic and repelled by the hydrophobic tails of the membrane. Small, nonpolar molecules like oxygen can easily diffuse across the cell membrane, all by themselves without channels or proteins. Uncharged polar molecules like water can diffuse if they are small, while large molecules make diffusion slower. Large uncharged polar molecules require proteins and channels because they are so big and the speed that they diffuse at cannot happen by itself. All channels are responsible for facilitated diffusion, also called passive transport. Moving a molecule against its gradient requires energy. The energy is usually ATP. These types of transportations are called active transport. Only specialized transporters called pumps can do this. Each organelle has its own set of membrane transport proteins used to move correct molecules into the correct organelles. Some transporters, like the glucose transporter, can move molecules in either direction at different times, guided by the gradient. This is different for charged molecules, where there are differences in charges inside and out of the cell, because usually the inside is negative, and that pulls in positive ions and repels negative ones. So, charged molecules contend with the push or pull of their concentration gradient, just as their electrical charge. Transport vesicles have two movements. One is from the ER to the Golgi, to other regions or cell membranes, and two, from cell membranes and inward to lysosomes, to the rest of the cell through the cytoplasm. The main role of transport vesicles are to carry soluble protein and membrane that are hydrophilic. Vesicle regulation and routing depends on proteins embedded in the vesicle membrane. Vesicle budding, the creation of vesicles, occurs due to protein coats. They are called coated vesicles. The coat holds its shape, targets the vesicle, and ensures proper contents inside. One coat type is a Clathrin coat, used for budding off the Golgi for secretion. Here's the Golgi. The inside is the lumen, and then this is the outside. Different adaptins are specific to proteins, and they secure the clathrin to the vesicle membrane. It chooses the cargo to load by gathering the cargo receptors with its protein bound to it. Cargo trapped by a receptor, and then the receptor is trapped by adaptin. Different adaptins in different membranes are each specific to a subset of receptors. Adaptins also gather Rab proteins, V snares, and call in the clathrin mesh. The clathrin protein surrounds its target protein at the Golgi. It will start pulling that cargo out of the Golgi. This spherical structure is called a clathrin-coated pit. Dynamin, a small protein, will then form a ring around the neck of the vesicle and pinches it to release the pit and to fully form this clathrin-coated vesicle. Vesicles are moved by walking proteins to get to their final destination. The vesicles and target membrane must recognize each other. This is possible because of Rab proteins, which are specialized proteins that tell its point of origin. Each membrane has its own subfamily of Rab. Additional recognition is done by the snare proteins. At the target membrane, there are tethering proteins. Tethering proteins recognize Rab and bind right above it. This holds the vesicle in place so it doesn't float away. The membrane also has T-snare proteins that interact with the V-snare proteins on the vesicle. This interaction stabilizes the docking process. The snares will bind and twist around each other until the vesicles touch the target membrane. Now fusion can occur where the vesicle and target membrane are touching. Before the coated vesicle fuses with the target, the clathrin coat sheds off. This allows the membrane of the target to fuse with the membrane of the vesicle. The cargo of the vesicle is then released. Fusion is finished when the vesicle becomes part of the target membrane and releases its contents. The cytoskeleton is an intricate network of protein filaments that extend throughout the cytoplasm of all eukaryotes. The three protein filaments that make up the network of the cytoskeleton are 1. Intermediate filaments, IFs, that is made of a family of proteins. 2 are microtubules, MTs, made up of tubulin proteins. And 3 are microfilaments, also called actin filaments, made up of actin proteins. IFs specialize in tensile strength. Their function is to withstand mechanical stress, such as pulling or stretching. They do this by stretching to distribute the forces of the mechanical stress throughout the entire cell. This mechanism prevents the cell from tearing. IFs are located between the MFs and MTs in the cytoplasm, where they form a network surrounding the nucleus and extending outward to the cell membrane. IFs are made of individual fibers that twist into strands. They are anchored on either side of the cell. They are also found in the nucleus, where the main component of the nuclear cortex is nuclear lamina. So, a pair of IF subunit proteins wrap around each other to form these dimers. Then two dimers will wrap around each other in a staggered orientation to form a tetramer. Those tetramers are then twisted together to form an IF in a rope-like structure. The result is a bundled, twisted network of individual strands, as you can see here. IFs are found in high numbers in cells experiencing most mechanical stress like neurons and muscle skin. The four classes of IFs are keratin filaments and epithelial cells located in the cytoplasm, where each keratin IF distributes stress of stretching. 2. Vimentin is in the connective tissue, muscle cells, and support cells of the nervous system. And 3. Neurofilaments are for structural support for neurons. These three are found in the cytoplasm. The fourth one is nuclear, and it's nuclear lamins that provide strength in the nuclear envelope of animal cells. Nuclear lamins form a two-dimensional mesh for stabilization. They must disassemble and reform with cell division, making them dynamic. This is controlled by phosphorylation to weaken the affinity with other lamins to cause disassembly. Phosphatase dephosphorylates to cause assembly. The function of microtubules is transport. MTs extend from the centrosome to the interior face of the cell membrane, creating protein tracks or highways for shipment. These highways are used by motor proteins for vesicle transport. MTs also anchor and localize organelles. Centrosomes cannot give the MTs a defined location. Therefore, it just builds MTs randomly in hopes that they will dock somewhere important, like at an organelle. In mitosis, MTs are disassembled and reassembled as mitotic spindles and sent to chromosomes to pull sister chromatids apart. Lastly, static MTs are the protein bases for components involved in cell crawling. They are hollow tubes of proteins that can rapidly disassemble at one location and assemble elsewhere. This is necessary because they have two main structures, which are microtubules and mitotic spindles. Dynamic instability allows for this rapid formation and destruction process. MTs have a point of origin called the centrosome. Lying on the outside face of the centrosome, gamma tubulin proteins are found in this ring formation. This ring formation serves as the starting point for MT formation. It's called the nucleation site. The subunit of MTs is tubulin proteins. They are found as dimers of two globular proteins. The dimers stack together to form a wall chain of globular spheres. It's about 13 protofilaments that will make an MT. They are organized as alpha beta from beginning to end. Because each protofilament faces the same direction, there is polarity where the side embedded to the centrosome is negative and the opposite side is positive. Tubulin dimers can be added to either end, but it prefers the positive end. Alpha beta heterodimers of tubulin proteins bind to GTP, which raises its affinity to form or interact with the microtubule. Tubulin proteins attached to GTP is in its stable form. Tubulin dimers have GTP-ACE. GTP-ACE cleaves GTP to GDP. GTP-ACE causes the dynamic instability because GDP-bound tubulin dimers are unstable and do not want to be bound on an MT. MT polymerization will continue, and as that happens, the GTP-ACE will begin to cleave its GTP to GDP. Tubulin protein attached to GDP is in its unstable state where its affinity for interacting with the microtubule is lower. This cleaving happens slowly as there is a time lag. During the time lag, MT polymerization is continuing to happen, which results in this GTP cap at the positive end. Although all the tubulin proteins on the negative side is unstable and does not want to be part of the MT, the GTP cap provides stability for the entire MT. Polymerization continues until either the microtubule has reached a target or there are no more tubulin proteins for polymerization. When it does reach a target, for example, the Golgi, anchoring proteins from its organelle will squeeze and hold the microtubules in place. Eventually, the microtubule will cleave all of its GTP to GDP, and there will be no GTP cap. But that's okay because the microtubule is anchored, so it is stable, and it is ready for a motor protein to walk along it for vesicle transport. Disassembly will happen for one of two reasons. The first reason is in mitosis. In this case, anchoring proteins will release the microtubules, and since they are all GDP bound, tubulin proteins will disassemble from the microtubules to its original individual dimer structure. From there, they will take up GTP and be ready to form into a microtubule again. But now they'll form into mitotic spindles, and that's dynamic instability, the ability to assemble and disassemble swiftly to provide different functions in the cell. Microtubules will also disassemble where they do not hit a target. The GTP cap will slow down as the number of GTP-bound tubulin proteins around decreases. This allows a time lag to catch up, and eventually there will not be a GTP cap, as all the proteins have cleaved GTP to GDP. Without the stabilizing forces, the microtubule will disassemble. Microfilaments are thin, flexible threads about 7 nanometers in diameter. They are made up of globular single monomers that come together to make a single filament. Actin subunits are wrapped and braided around each other to form a tightly compact, thin filament that is shorter in length than microtubules. These microfilaments contain polarity, such as microtubules do. They have associated proteins that bind to actin filaments to stabilize them. Microfilaments exist in bundled clusters to make up actin-based structures in the cell. Microfilaments are made of actin monomer subunits. Each monomer is bound to ATP. ATP-bound actin monomers have an affinity to attach and grow to microfilaments on the positive end. Once a monomer attaches to a microfilament, the ATP is cleaved to ADP at the negative end. And those monomers have lesser affinity for being part of the filament, so they disassemble from the negative end. They must be dynamic to have an ease of assembly and disassembly to carry out their cell functions, as we'll get to in a little bit. The cytosolic face of the membrane is where actin concentration is the most high. They are linked into a protein meshwork by binding proteins for mechanical strength to the membrane, and it helps dictate the shape of the cell and its dynamics. Cell crawling is a 3-step process. First process is the cell pushes protrusions at its leading edge. The cell forms a sheet-like structure called lamellipodium. It contains a dense actin meshwork that assembles at the leading edge of the cell. Lamellipodium reaches out by pushing against the base of the membrane. This is driven by new actin polymerization at the positive end of the filament. Step 2 is anchoring. And the finger-like protrusions adhere to the extracellular surface on which the cell is crawling towards, reaching an anchoring point. These protrusions are filopodia, and that contains small bundles of actin filaments with its positive end facing outward. They linearly push along the extracellular surface. Filopodium sticks to the anchor point using integrins. Integrins is a transmembrane protein that serves as a protein bridge to connect the extracellular membrane, the ECM, to the cell membrane. Integrins will attach the actin inside the cell and the ECM. This allows the cells to stick to the surface it is trying to reach. Step 3 is pulling the entire cell forward towards that anchoring point. Here, actin in the cell cortex is used. Myosin is located, which are actin-binding proteins here. They provide energy for movement. Thick filaments are myosin molecules self-assembled into thick bipolar bundles with head domains on both sides. Each head associates with two actin filaments. Each head domain interacts with actin, providing the force for the slide motion. Myosin can be bound to either ATP or ADP. Myosin bounded by ADP increases its affinity for actin, and myosin bound by ATP decreases its affinity and makes myosin let go of actin. Myosin is bound to actin when it's bound to ADP, but myosin has a high affinity for ATP now. So as ATP attaches to myosin, myosin will let go of actin, and then the ATP will be cleaved to ADP plus P and rebind to actin. That extra phosphate group will unbind from myosin, causing the myosin head to shift forward, and that drives the cell to push forward. Rho protein serves as a mediator for actin. All of the signal cues talk to Rho, and then Rho will give actin a summative signal to either crawl or not crawl. Cells must sense and respond to the environment and to other cells. Signal transduction is the process of converting cell signals from one form to another. Signaling cells are cells that send the information and release a signaling molecule. Signaling molecules move and are detected by a receptor on a target cell. And three styles of cell signaling are 1, publicly, 2, with limited targets, or 3, where they can be extremely private. Receptors can only bind a single signal molecule, and they relay the message by mechanical form changing, where they change shape when bound by a signaling molecule. The signal and receptor complex is the primary messenger. Relaying the messenger to a second messenger happens when signal transductors pass on the message from one intracellular signaling molecule to another. They create a signaling cascade, which is a simple relay of signal from outside of the cell to the inside to amplify the signal received. Crosstalk occurs when multiple signals can be integrated to create new signals. These signals are modulated. One intracellular signal can affect multiple cellular pathways, launching a complex cellular response. Crosstalk allows cells to specialize their response to a given signal. Cell surface receptors are embedded in the cell membrane and bind to extracellular signaling molecules. The receptor transduces the signal into the cell. The end results are a response from the cell. G protein coupled receptors, or GCPRs, carry the signal into the cell by a series of modified proteins. And here's the process. A signaling molecule binds to GCPR, and this causes the receptor change shape, and the shape changes activities of the G protein. That G protein is associated with the receptor at the cytosol face of the membrane. It is made of 3 subunits: alpha, beta, gamma. G proteins can bind to GTP and GDP. When G protein is inactive, the alpha subunit binds to GDP. To turn G protein on, a signal molecule binds to the receptor and the receptor changes shape. The protein is bound to GDP, and the change shape decreases G protein's affinity for GDP and increases its affinity for GTP. The G protein will let go of GDP and grab GTP to activate the G protein. G protein has an enzyme subunit, GTPase, that cleaves GTP to GDP to inactivate G protein. Function of the G protein is one to activate to open membrane-bound enzymes or channels. This is a GCPR controlled enzyme where ion channels will open. 2 is affecting enzymes. Enzymes are activated by G proteins. An example is adenylate cyclase, where ATP is converted to CAMP and phospholipase C. CAMP activates protein kinase, and kinase A phosphorylates molecules to turn on or off target proteins. The steps are 1. A signaling molecule will bind to a receptor, and the receptor activates, causing it to activate adenylate cyclase. When adenyl cyclase is turned on, it modifies many ATP to become many CAMPs, and CAMP binds to many protein kinase A's, also referred to as PKAs, and activates it. The PKA move to their target protein and phosphorylates them. This phosphorylation will either turn the protein on or off. One extracellular signal has caused a cascade of cellular response in the cell. This is amplification, where one signal changes the cell's behavior accordingly. For phospholipase C, calcium serves as a second messenger and uses membrane components, PIP2. A signaling molecule binding to GPCR activates G protein, which activates phospholipase C, or PLC. When active, it hydrolyzes PIP2 into 2 other molecules. IP3 is the second messenger, and DAG remains in the membrane and activates protein kinase C. Kinase C phosphorylates target proteins such as calcium channels. It allows some calcium into the cell from the membrane. IP3 goes to the smooth ER to open channels. It triggers the release of calcium. Calcium binds to PKC to activate it to protein calmodium to become active. Calmodium binds to CAM kinase to cause a cellular response. Turning CAM kinase on phosphorylates many proteins. Phosphorylation will either turn the protein on or off according to its initial state. Those cells being expressed or repressed cause a change in the cellular behavior. The cell cycle control system is the network proteins monitoring and signaling the readiness of all the cellular components for all the stages in the cell cycle. G1 and G2 are preparatory stages and also where the checkpoints happen. S phase is the DNA synthesis stage, and M phase is for mitosis and cytokinesis. The checkpoints occur at G1 to confirm cellular environmental favoring. At G2, it confirms the completion of DNA replication and repair. And in the M phase, it confirms mitotic spindles are attached to sister chromatids in anaphase. Cyclically activated kinases are key proteins turned on and off in pattern to achieve the cellular effect. Kinases for all stages of the cell cycle are present at all times. The molecular toggle switch is the phosphorylation cycle of 2 proteins. Kinases for phosphorylation and phosphatases for dephosphorylation. Cyclins bind to cyclin-dependent kinases, CDKs, to control them by turning it on or off. The type of cyclins made depends on the stage of the cell cycle. Each stage of the cell cycle is a set of cellular behavior according to its function. To change the cell's behavior and proceed to the next stage, you need kinases. So kinases will change the cell's behavior. Kinases trigger different cell cycle stages through the phosphorylation of proteins. But in order for them to activate, cyclins are needed. Cyclins bind to kinases, and this binding activates the kinases, which will phosphorylate and turn on proteins or turn them off, which will lead to a change in cellular behavior. When a specific cyclin is made, it will bind to activate a CDK. The CDK will phosphorylate target proteins for cellular behavioral changes. Active CDKs will induce the expression of the proteins necessary to continue the next step in the cell cycle. For example, if the next step is DNA replication, the proteins that CDK will trigger are the proteins to ready the cell for DNA replication, such as DNA polymerase, helicase, etc. CDK inhibitor proteins block interactions between cyclin and CDKs to stop CDKs from being activated. This is a checkpoint solution. These checkpoints detect DNA damage in G1 and S phase to prevent DNA damage is present. Without a certain cell stage cyclin binding to the same cell stage kinase, the cell will not get the signal that the next stage of the cell cycle needs to begin, and therefore the cell will not behave accordingly and will not proceed in its cell cycle. There are 5 cell junction types that hold epithelial cells in place to form epithelial sheets. Epithelial sheets are fundamentally a single sheet of cells. They can be stratified sheets of cells that form thick sheeted tissues. There are 5 morphologies: the simple, stratified, columnar, cuboidal, and squamous. Their function can be for either a single purpose, diverse purpose, secreting, absorption, or senses. It is the only cell type that faces the inside and the outside of the human body because they can lay as sheets. They act as cellular receptors that can hear and pass the information inward. Epithelial sheets are polarized and rest on the basal lamina. Tight junctions seal cells together so nothing can leak in between them. Claudin and occludin proteins arrange like two sides of a zipper and zip up to seal the cell together. It prevents molecules from leaking and defines the membrane domain. For example, it holds skin together so that when water gets on our skin, it does not leak through. Without tight junctions, we would swell up with water and burst when showering or in a pool. And who doesn't like going for a good swim on a hot day? Tight junctions are also found in the gut in its lining cells. Without tight junctions, whenever we eat food, all of the nutrients would go to the other tissues and never be absorbed. And so, therefore, we wouldn't be able to live. These tight junctions serve as a barrier so that nothing leaks out to where it doesn't belong and nothing sneaks in to where it's not supposed to be. Numbers 2 through 4 are adhering junctions and desmosomes. They bind one epithelial cell to another. Hemidesmosomes bind an epithelial cell to the basal lamina. All 3 provide sheets of epithelial sheets with mechanical strength by joining individual cytoskeleton together. Adherin and desmosomes use cadherin, a transmembrane protein for cell connection. The C stands for calcium used in this interaction. Each cell has a cadherin binded and 2 cells nearby will interact through the cadherin. Actin filaments, or keratin, are tethered to cadherins through a linker protein. This links cadherins directly to the cytoskeleton. The linkage allows connecting cells to feel each other's movement through these actin filaments that are woven between all of the cells. Hemidesmosomes use integrins to link collagen to the basal lamina. Number 5 is gap junctions. They're used for private communications and making openings. Matched membrane complexes, called connexons, meet between 2 cells and create a continuous channel across the 2 cell membranes when the 2 meet and then line up. In living systems, almost every tissue is an intricate mixture of many different cell types that work together to sustain life in a complex supporting apparatus. Cancer cells proliferate, invade, and metastasize. Cancer cells break the rules of healthy cells. Those rules are dividing only when needed, live for as long as needed, undergo apoptosis when needed, maintain cellular and biochemical characteristics, and just stay put in their respective place. When cells break these rules, they form a mass of rapidly dividing selfish cells. The mass crowds, pushes, and destroys the tissues in the way. Its official definition is a relentlessly expanding clone of abnormal cells. That's a cancer cell. Cancer is caused by an accumulation of very specific mutations such as deactivating DNA repair enzymes, DNA proofreading, bypass, or cell cycle checkpoints. There are four pathways of mutations causing cancers. 1. Cell proliferation. 2. Control of cell growth. 3. Response to DNA damage. And 4. Response to cellular stress. The genes mutated are all different, but these pathways are affected in almost all human cancers, where under and overactivity of the pathway causes cancer. Mutations can make supercells. Their properties are rapid division, invasiveness, and attract blood supply, degrade the ECM, and enter the blood or lymph system. Healthy, normal cells follow all these rules for the greater good, which is the organism. This includes having to undergo apoptosis when needed. For example, when the cell cannot repair its DNA damage, there's too many mutations. But if one sociopathic cell does not follow the rules, it will become cancerous, and that cancer will spread to other cells. These mutations are the reason cells become rogue cancer cells. Cancer cells are defined by two main properties. 1. They proliferate in defiance of normal constraints. And 2. They invade and colonize regions reserved for other cell types. A benign tumor only has the first property and can be effectively removed with surgery. The faster it is found, the easier it is to remove and the less cancerous cells have formed. Malignant cancer has the capabilities to invade surrounding tissues, divide, and spread. Metastatic cancer has the two properties and invades the circulatory system, including the blood or lymph system. It carries cancer cells anywhere in the body and gives rise to secondary tumors. These tumors retain properties of the primary tumors as they contain the original physiology and molecular hallmarks. Properties of cancer cells are 1. Cancer cells don't need signals from other cells to thrive. For example, RAS binds to GDP when active and GDP when inactive. A mutation on RAS will result in always being active, and the cell will continue to divide because it thinks it's receiving the signal to do so. 2. It does not default to apoptosis. Normal cells sacrifice themselves for the greater good, which is the organism. And in this case, with no p53 being made when cell damage is noticed, then no DNA damage is sensed and no opportunities for repair, and also no opportunities for apoptosis. And 3, cancer cells are immortal and never cease dividing. Genetically, they are unstable with chromosome mutations. And 5, they are abnormally invasive. There is a lack of cadherin and proteins needed for cell junctions. The lack of adhesion proteins allow the cells to slip away and metastasize. Lastly, 6, cancer cells can adhere to, survive at, and divide in foreign tissues of other cell types. This is the definition of metastasize. Normal cells would normally die when they're misplaced because of the extracellular cues, and they would default to apoptosis. That's the end of my e-report. It's been a great semester. Thanks for listening.